You got an okay today?
I did. Wait, do you mean the physically or the Teams meeting?
In the Teams meeting.
Oh, okay.
I
So does
think
someone,
last month, wasn't there an issue?
yeah, I think like people had to be let in
Yeah, okay.
last time.
I think I got it set up correctly this time, obviously, so.
Yeah.
Yeah.
Um, what was I gonna say? So I was
And you-
looking on the the invite and I didn't see any. I guess we have a talk in July, but I didn't see that on the calendar, but I can look again.
Did we
I
get
suppose I
somebody for July? I didn't think
don't
we
know.
had somebody.
That's what Moromi said, I guess. But I was,
I'll have to look through my emails. Okay.
yeah. But we already have someone for July, and I was like, I don't know who.
I'll look. And then I'll
I
send
was
out
like, I didn't see
and then
anything
I'll
on the
send out an
calendar.
invite. There's a lot going on.
Um,
So, okay. Anybody in the waiting room, or are we okay?
I don't see anybody in the waiting room.
And you can admit them from the waiting room?
I don't know.
Oh, okay.
Uh,
I will hang around for a little bit.
yeah. Hi.
Okay, sorry, I had to find out. We do not use Teams much down here in Maryland, and I couldn't even figure out how to unmute myself for a second. This is a new thing for you guys, right?
Yes, yeah.
Rebecca, you look like you're in like Chicago or something.
It's a virtual background, but
Oh, okay.
I find it nicer than my walls.
Okay. All right. All right. Well, thank you for taking over, so to speak, today. It has been I couldn't figure out what's going on, but apparently it's because it's Friday the 13th. I don't know.
Oh, it is.
Oh, I forgot about that.
I'm not normally superstitious, but the day's been so wonky. I was just in a meeting with someone. I was like, what is going on? And she said, it's Friday the 13th. And I thought, okay, that explains literally everything since this morning.
Really funny.
So
So I'm going now with some friends for dinner tonight, and I was actually supposed to go out with the same friends on March 13, 2020, and they just reminded me that that's when everything shut it down because
oh
of
God,
COVID.
Wow.
So yeah, so we're supposed to have this same dinner, the six of us, on March 13th, 2020, and they're like, "Do you remember when all these schools shut it down for COVID?" Because we
Yeah.
all
Oh my
had
God.
kids in the same class.
Oh geez, that doesn't bode well. Well, hopefully that goes well.
Hello. You are muted. I'm not sure if you meant to be muted.
Me?
No, sorry, Dr. Dlugos.
Oh,
Now you're
hello,
not
good
muted.
afternoon. Can you
Yeah,
hear
hi.
me now?
Yes,
Yeah.
we can.
All
I'll
right.
be introducing you, but if you wanted to practice, like share the screen just to make sure things work on your end, let us know.
Sure, I can do that.
I mean, we have several minutes. Yes,
How's that? Can you see it?
looks great. Yeah.
And can you see the pointer as well?
Ooh, yes.
All right, great. So I'll stop sharing.
Okay.
Or do you want to leave this up just so it's ready to go? Or
Yeah, you can do that. I'll just
All
send the CME code.
boy, people know they're in the right place.
Yeah.
right, exactly. we're a little, Dennis, I probably, and this is always pretty well attended. We are the Friday before some people's spring break. So, meaning children, not faculty.
Uh, yes. Well, that's actually why I'm available, because it's the Penn Medical School spring break this week. And I always have a Friday 1 p.m. conflict, except this week. So,
yeah, I forgot. Did we have spring break in medical school? I don't remember that.
I don't think we did.
just the first years.
Oh, I don't remember that at all, but
I remember winter break. And I remember people telling me, enjoy winter break. This will be
Right.
the last time you get winter break.
right. That is true.
And then at NYU, Steve Galetta would just close the clinics between the week of Christmas and New Year's. And so the faculty would kind of get a spring break.
I find that that's often like a lot of time when people want to come in. Like there's all the so like the teenagers off or college students off, but yeah, it's a mixed bag. I think you're right. And we're just pinging our group here too. I think people are just, it looks like just kind of coming off of clinics. So we may want to just, Rebecca, give them just a few minutes, but
Yeah.
All right, I'll be right back.
How big is the Maryland med school class these days?
oh God, you know what?
Do you know?
I really don't.
As soon as I started asking that, I'm like, I don't know if you would know.
Yeah, I'm not in that world so much. I know that I do know that they are enlarging it, because there's been a lot of, I hear the downstream conversations of when they're talking about, okay, you know, as they're enlarging the class, there's going to be more people coming through for rotations, and, you know, how do we accommodate that, et cetera, et cetera. But I actually don't know.
Mhm.
At Jefferson, it's like 250.
Jefferson's very
Ish.
big. I graduated from Jeff in 1997, and we were 235, and we were one of the largest medical school classes at that time. I remember if we actually all went to class, there weren't enough seats in the auditorium.
Jefferson's now over 290.
Oh my God, is it, Mike? Wow.
Yeah, they're in the hanging from the rafters on swings. They've taken the advantage of that nobody shows up for lectures anymore.
Right.
To have everything online, you can kind of ask questions, and you know, of the 290, maybe 40 will actually show up in a room.
Oh my gosh.
Yeah. And hello, Dennis.
Hello, Mike.
Yeah.
Good to see you.
Yeah, good to see you.
Hey,
My
Dennis.
my apologies in advance. I have to leave. I actually have a doctor's appointment at 1:30. So
All right,
I'll
no
be here
problem.
part of the time.
Poor excuse, Mike.
Yeah, well,
I'm kidding.
it's not easy to get an appointment these days. Take it
I
when you
know.
can.
15 minutes, 15 minutes for a follow-up, Mike. Come on, you know that.
And I'm in good health too.
And you're in good health, exactly, yeah. Hey, Dennis, how's it going?
Hello, Pete. I'm doing well. How are you?
Good to see you.
Same.
Rebecca, it's up to you, but I see
We have
a nice-
21 people.
we
I
have a
think we
nice
can
attendance,
start.
and people
Yeah.
are joining by the moment, so you could probably get started.
Yes, and I'm gonna
Yeah.
just once I after the intro, I will resend the QR and CME code. All right, happy Friday the 13th, everybody. It's my pleasure to introduce Dr. Dennis Dlugos. He is a professor of neurology and pediatrics in the Perelman School of Medicine at the University of Pennsylvania, and holds the Tristram Colquitt Jr. Endowed Chair in Pediatric Neurology. He's the director of the Pediatric Epilepsy Program at Children's Hospital of Philadelphia, where he also serves as the director of the section of clinical neurophysiology and the vice president of the Epilepsy Study Consortium. And clinical research interests vary from clinical trials, epilepsy genetics, and epilepsy surgery. And looking forward to having him talk to us about outcomes in epilepsy trials.
All right. Good afternoon, everybody. Thanks so much for the invitation. I usually have a conflict in this time slot, but as we were saying, Penn Medical School is on spring break this week, so my Friday afternoon is available. So I'm very happy to join. So we're going to talk about outcomes in both trials and practice, and hopefully the two can inform each other. So my disclosures, I have research support from the Epilepsy Study Consortium, and I'll make some references to the consortium throughout the talk. So here's what we're going to talk about. We're going to do a quick overview of epilepsy trial outcomes ranging from natural history studies, observational studies, to interventional trials, including the emerging precision therapy trials. And as we think about the outcomes in those trials, we'll reflect on what it means for outcomes in clinical care. And along the way, I want to talk about the use and sometimes misuse of the term treatment resistance, because that's certainly an important phrase in our field, and it can lead to various branch points in care, but always with what outcome are we thinking about when we try to help patients with treatment-resistant epilepsy. Okay, so this is a picture from a book on evidence-based medicine. And why do we have to do these trials? Well, these are the alternatives. Eminence-based medicine, vehemence-based medicine, eloquence-based medicine, providence, diffidence, nervousness, and confidence-based medicine. All of which, if you read the descriptions on the right, we live in this space a lot. Some of us are guilty of these things. And this is not great. We can do better than this, and we have, but we have a long way to go. So every trial I will show you is deeply flawed, but all of these alternatives are perhaps even more flawed. So let's start with observational studies, natural history trial. So this is recent. It's from Scandinavia using the almost, I guess, current ILAE classification system regarding seizure types, epilepsy, and etiology. And this focused on pediatrics. And I think it's helpful to just take a step back with a population-based cohort and see how our patients do. So over 1,000 patients in this trial. And they had 10-year outcome data, median follow-up of 10.6 years. So no surprise, 1 in 100 children had epilepsy. That's kind of the standard definition. Maybe better than we might think, seizure freedom attained in 78%. Higher than I might have expected. I usually say 60 to 70. So this was better. Now, of course, this is population-based, so you get everyone. A key table in this study was this, where they looked at the proportion of patients who were seizure-free at 1, 2, 5, and 10 years since onset. And you see, if you look at epilepsy type, the numbers look good, except for patients with combined focal and generalized epilepsy. 10 years seizure freedom rate of 16%, much, much less than anyone else. And I want to highlight this in part because we see this all the time in pediatrics, certainly at CHOP, kids with focal seizures and generalized seizures. It's been officially recognized in the ILE classification since 2017. So it's not that we can't or won't find the focus. They have both for whatever etiology, whatever mechanisms. So combined epilepsy does particularly not well in terms of seizure freedom, but other types in a population-based sample do pretty well. The other thing that surprised me was when you look at syndromes in groups, they use the still elusive term DEE, developmental and epileptic encephalopathy. So kids with developmental delay, slower disorganized EEG and some seizure types, and they're viewed as a very treatment-resistant group, but 10-year follow-up, 45% were seizure-free. That's not great, but it's better than at least I would have predicted. So I just wanted to start with this to sort of lay the landscape for a population-based cohort. So etiology in 2025, and I think future trials will look more and more like this. So this is how a contemporary etiological breakdown goes. Still 40% unknown, and then look at the various subdivisions of genetics, syndromes, both developmental and structural malformations. So I think it's helpful to have data like this on a broad scale so we can get a sense of the diversity of the epilepsies. It's not particularly surprising of a list. Maybe it's a little humbling that despite top flight genetics and imaging, 40% remain unknown. All right, so now let's talk about therapeutic trials. And we're going to start at the beginning with new onset seizures or epilepsy, and we'll work our way through most of this list. We're actually not going to talk about seizure rescue trials, status trials, or neonatal trials, because there's plenty to talk about just with the first three topics. So infantile spasms. I think there's, even if you don't see patients with infantile spasms, I think there's an important point for the next few slides. So the infantile spasms world made a fundamental advance in 1996 when this randomized trial was published in Pediatrics. High-dose ACTH versus what we would now consider low-dose prednisone for infantile spasms. Since the ACTH was an injection and the prednisone was oral, it was single-blinded. The outcome raters, which was an EEG outcome in part, obviously the families were not blinded. They knew if their child got an injection or was taking prednisone. And in this trial, high-dose ACTH overwhelmingly beat low-dose prednisone. 80% resolution of spasms and hypsarrhythmia versus 29%. So this sort of enshrined ACTH as a drug of choice for new onset spasms. A few years later, 1999, this study doesn't get a lot of attention because looking back, maybe it wouldn't be okay now, but this trial was fundamentally important in bringing vigabatrin as a first-line treatment. What did they do? Well, it was prospective, randomized, and placebo-controlled. 40 children diagnosed with newly diagnosed spasms received vigabatrin or placebo for 5 days. And after 5 days, the vigabatrin group had a significant decrease in spasms and improvement in EEG. So with a 5-day placebo arm, vigabatrin demonstrated at least short-term efficacy in the treatment of infantile spasms. Could we do this today for new onset spasms? No, we probably couldn't, or at least it would be a struggle. So after these trials, which are old now, ACTH, vigabatrin, and then through other trials, not regulatory trials, high-dose prednisone became the standard first-line treatments for infantile spasms. And they're okay, but they're all very flawed. Side effects, they don't work for everybody. Relapses are a big problem. And strong statements were made by pediatric epileptologists about any new trial of infantile spasms, new onset spasms, should use one of these treatments in all of the arms, because it would be unethical to randomize patients to something other than these gold standard therapies, hormonal therapy or vigabatrin, which completely crippled advancing the field. We couldn't make progress. You couldn't do studies that anyone would do because you weren't going to beat the efficacy of these treatments, even though they were flawed. The outcomes in infantile spasms trials are resolution of spasms, so complete resolution, and normalization of hypsarrhythmia at 4 weeks. These short-term trials didn't consider relapses. So we were stuck. until some, I think, very clever investigators from India said, you know what? These treatments are all flawed. We think we can do better. So this was published in 2025. It didn't get enough attention, I think. They compared ketogenic diet versus ACTH in new onset infantile spasms. So a standard treatment versus ketogenic diet. So they disregarded the advice that you couldn't do this. And you know what? Keto diet beat ACTH. If you look at median percentage spasm reduction here, you see ACTH here, 66%. Keto diet, 76%. It's not a statistically significant difference, but certainly keto diet was no worse and trended toward better spasm reduction. Same if you mix in improvement in EEG, and even better, keto diet had fewer relapses. So this has not really been integrated in practice at least at my center yet. We are certainly a large keto diet center, but we're not doing keto diet for new onset spasms. But I think there's evidence that you can, and it may actually be better. So the reason why I bring that up I realize it's a mixed audience, is there was strong statements that we couldn't do this type of trial. And we could, and we did, and maybe it's going to make our treatments even better. So I think we have to sort of think outside the box somehow if we're just stuck, and look at some novel trial designs. So what about good old-fashioned focal onset seizures? So we're all influenced by this trial from a long time ago. I want to bring it up again to sort of go back a little bit. This was oxcarbazepine when it was being developed. Kids and adults with untreated focal onset seizures, not single seizures. They all had at least 2 seizures, and randomization to oxcarb or placebo. Notice the package insert is the reference. That's not like I was too lazy to look up the real reference. It actually was never published in the peer-reviewed literature because it was viewed as, you can't really do this. Randomizing kids and adults for 80, 90, 100 days to placebo, despite having 2 or more seizures. So maybe no surprise, oxcarbazepine was better than placebo. But know that not that long ago, we could do this type of trial. We don't, or at least we did. Whether we should have is a different question, but we don't do it anymore. So we just have to face facts with new onset seizures, especially in adults. It is hard to do any sort of placebo trial. You could do high dose, low dose. It's been done a couple times. This is most recent, and it's an interesting trial. This is topiramate. So new onset, focal onset seizures, 400 milligrams a day versus 50 milligrams a day. They're trying to find the full dosing range, and there was data that 50 milligrams a day was as effective as the other drugs being used at the time. So in this trial, this is the Kaplan-Meier curve with breakthrough seizures. So higher on the curve means fewer breakthrough seizures. Maybe no surprise, high dose topiramate beat low dose topiramate, but still roughly 50% of patients going out to 500 days were seizure-free on low-dose topiramate. So I think a message there is, as we incorporate new drugs or new treatments, all of them have side effects. The trials are the trials. They're like fixed doses. So trying lower doses can be a good strategy. And you might get seizure control for many patients with presumably better tolerability. So a quick detour to another new onset topic, absence. Again, older data, ethosuximide versus valproate versus lamotrigine in childhood absence epilepsy. You probably know that this study reconfirmed that ethosuximide was the drug of choice, and ethosuximide and valproate were both more effective than lamotrigine. I bring this up because the outcome measure was quite specific and I think has been misinterpreted and misapplied. So this trial was published in 2010, and the outcome was basically, so treatment success, which was the outcome, no reported seizures, no intolerable seizure medicine side effects, and no generalized spike wave discharges longer than 3 seconds on the 1 hour waking EEG. That was the EEG component of the clinical trial outcome. And that's all it was. It was an outcome chosen for a trial because we had to pick something. There was data that says most patients have some impairment of response at 3 seconds. So if the discharge was longer than 3 seconds, it was considered a seizure. It was not meant to be a statement about clinical practice in any way. So I was somewhat surprised when earlier this year, colleagues of mine wrote that it was this New England Journal study that established the 3-second rule as the standard for future clinical practice. And I read that and I said, what? No, it was not meant to establish anything for future clinical practice. It was a trial outcome. And it's a whole nother topic that we have dabbled with our pediatric and adult colleagues at CHOP and Penn about how long of a spike wave discharge is definitely a seizure or is possibly a seizure. But for any of you that were raised that the 3-second rule is standard for practice, no, it is not the case. It was a trial outcome. So sometimes trial outcomes and clinical practice can actually misinform each other. All right, so another new onset from the old days, great ideas that never happened. So let's study levetiracetam versus oxcar versus lamotrigine in kids with new onset epilepsy and look at cognitive and behavioral outcomes. Great idea. And you see, terminated, poor recruitment, never happened. Budget was not great. Families didn't like that it was randomized but not blinded. So basically the computer picked the drug, but everybody knew what the computer picked. And that just didn't sit well with families. They were almost better off not knowing, like randomized double blind rather than randomized but unblinded. But for a variety of reasons, we don't know what is the preferred first ASM in children or in adults or adolescents for that matter. It was tried and it didn't work. Okay. Refractory focal onset seizures. This by design, this has no label because pick your drug, pick your trial. You could put in most of our drugs and the active drug will reduce seizures by 50, 60% and placebo 20%, and you'll get an approval. So lots and lots of refractory focal onset seizure trials, and they continue to happen using median percent seizure reduction. Well, so not so recent, but not everybody knows this anymore. Recent FDA changes within like the last 5 years is the newest drugs for focal onset seizures no longer have a monotherapy versus adjunctive therapy distinction. They're just approved for focal onset seizures, which I think is helpful for prescribers and patients. The other thing that sounds like regulatory small print, but is really important on the pediatric side, is what's called extrapolation of adult efficacy data. So if a drug for focal onset seizures only is shown to be efficacious and safe and tolerated, so gets an approval in adults, it is assumed to have efficacy down to 1 month of age. 1 month. So when does adult focal onset seizures begin? 1 month of age. Per the FDA, based on various biological reasons we can talk about. So if you have a drug approved for adults and focal onset seizures, you don't have to do efficacy trials in kids anymore. You do have to do safety, tolerability, and PK trials to get some data about the dose. but you don't have to show efficacy in kids. They are assumed to be efficacious. This only applies to focal onset seizures, does not apply to Lennox-Gastaut or any generalized seizure type, so only focal onset seizures. And this has real implications. Lacosamide and brivaracetam didn't do efficacy trials in kids, but they have approvals down to 1 month of age. 1 month of age, and other sponsors are taking this this route to a pediatric indication. Very quickly about Lennox-Gastaut, it seems like every trial is positive, except no. Negative trials do occur. This is the soticlestat story. Phase 2 data was encouraging enough to do a worldwide phase 3 trial. And here's the primary endpoint. They use what they call major motor drop seizures. And this is kind of a stunning curve. Placebo, 7% reduction in seizures. Soticlestat, 7% reduction in seizures. A total wash. It did nothing compared to placebo. I show this because sometimes we do nothing in visits because we're stuck. Too many seizures, too many side effects. Do you go up? Do you go down? Sometimes we just sit there like a placebo arm for 3 months until the next visit. And we all know sometimes things don't get worse. They stay the same. This was a minor decrease, but no difference. So sometimes with treatment-resistant seizures, doing nothing isn't worse than at least in this case a drug that was ineffective. So soticlestat will not be on the market anytime soon, and the Dravet trial looked a little better, but still was negative. So I think it is important to know that negative trials do occur, and there's lessons to be learned from all of the negative trials. Okay, now let me detour into treatment resistance. And I'm going to use the Lennox-Gastaut syndrome as the example. This was a review paper published last year, and I think none of this will surprise any of you, but it's going to set up the next few slides. So, all right, drug-resistant Lennox-Gastaut, or any type of epilepsy. Seizures unresponsive to at least 2 trials of adequate doses of appropriate anti-seizure medicine. All right, we're all used to that. I think the key thing is when a patient fits in this category, it does not mean they must have surgery. They should be considered for it, but there are 4 options: optimization of anti-seizure regimen, or neuromodulation, or resection or disconnection, if that's appropriate, or dietary therapy. I think the world in which we live is, so treatment resistance means referral to an epilepsy center. Well, I think everybody on this call is already at an epilepsy center. So any of our patients who are treatment resistant, they already are where they need to be. Treatment resistance doesn't mean do surgery. It doesn't mean put in a device. It means think about ASMs, neuromodulation, disconnection, resection, or dietary therapy. So you all know the neuromodulation options. And this was a paper that popped up last year that I think is very important for us in pediatrics. Maybe less so in adults because you have clearer indications, but it's an interesting title: Normalization of Deviance in Neuromodulation for Epilepsy. So what does that mean? What it means is normalization of deviance means when non-standard care becomes standard. And we're at a cusp of that happening, certainly in pediatric epilepsy. So our neuromodulation devices, DBS, RNS, VNS, they have their indications. You all know for DBS, it's adults. RNS is adults, VNS is approved down to age 4. And there are all of these off-label indications, both with age, children, adolescents, and location of the stimuli, of the electrodes. So there's a lot of off-label applications. So what's the big deal with off-label? We use drugs off-label all the time. Of course we do. And they are typically highly reversible. The drug doesn't work, it's not tolerated, we stop it. We have not recently had serious irreversible safety adverse effects from our drugs. That can always happen. But off-label drug prescribing is pretty reversible, where an implantable device is less reversible. So what the authors suggested, if you are dabbling in off-label applications, is that you think about it like this. So first of all, think about why you're doing off-label off-label stuff. It was a very sobering paper. Are you doing off-label neuromodulation because of prestige, because of career ambitions, funding pressures, etc., or not? We do it because it's the best option sometimes, because there are no other options. But just think about these things. And then, so an off-label device, you are deviating from standard practice. Things go fine. There is no harm. This is a feedback loop. So then you keep deviating because there's no perceived harm. But that can only go so far because we haven't really tested these things. So their recommendations are not to stop doing off-label neuromodulation, but to think about actually putting extra safeguards in, such as running it through your IRB. If nothing else, centralize outcomes, like carefully track your outcomes. And this, I think, is the practical point here, is emphasize safety, which is it's a buzzword these days, but I think it has a very particular meaning right now. So safety. So the way neuromodulation is thought to work, it might be 7 years at least before you see the full benefit. But there are safety metrics that can be measured all along, and every center, their advice, at least know what your safety outcomes are, even if you are waiting year after year after year for your efficacy outcomes. So some of the surgical neuromodulation trials are clearer than others about the problems with open label data of any type. This applies to drugs as well, but we're kind of in the neuromodulation phase here. So obviously there is bias, and there's all kinds of problems without a control group, blinding or randomization. Duration of follow-up is all over the place. Other treatment changes are happening. Put in a device, great, but someone's adjusting meds, hopefully, as well. And then what are the safety and adverse events? And 3 that are readily available for neuromodulation are return to the operating room, explants, or infections. And we'll come back to some recently published data on that in a second. So this was published last year as well. It comes from the Human Epilepsy Project #2 study, which was a observational cross-sectional study of patients with treatment-resistant focal epilepsy. So patients who might get neuromodulation or optimized ASM regimens, some surgery, things like that. And they were followed for up to 3 years. And the good news is that over those 3 years, 69% of the patients actually had seizure reductions based on their care, which was done at centers like are on this call. An interesting point was there was no difference in seizure reduction based on device or medication changes. So the news is like newsflash. Neuromodulation doesn't typically lead to seizure freedom. It can lead to reduction in seizures. The other newsflash is optimizing ASM regimens can lead to seizure reduction as well. We may not get seizure freedom, but both can lead to seizure reduction. So they basically say that treatment of focal treatment-resistant epilepsy improves over time, that ASM additions contributed to seizure reductions, and device-treated participants exhibited similar seizure trajectories to those without devices. Now, all of this has all the biases of observational, uncontrolled data, but it just goes back to treatment resistance means think of these 4 boxes: ASM, neuromod, resection disconnection, or diet. It is not a superhighway to neuromodulation, although neuromodulation seems to be a great option for some patients. And now some safety data. It's hard to find this in the literature. This is from a pediatric site published last year. 54 patients. So these were almost all off-label DBS or RNS. A few were above 18, but most of them were off-label patients. So 54 patients, and these were the post-operative complications. Their 4 RNS patients either had to return to surgery to be explanted, or had an infection, or or had a some wound-related complication, and 2 DBSs had some sort of immediate surgical complication. So this is 6 patients of 54, so I'm going to call that 10%. So this is early in the pediatric experience, but if we have in pediatrics a 10% return to surgery, explant, or infection rate, that's patients need to know that. And maybe another drug, also deserves to be in the discussion. It's possible all of your centers may have this data at your fingertips. We found ours. It was harder than I expected. It's there. But at least as we talk about neuromodulation in our surgical conferences, what we're trying to do is, all right, any updates to our safety data? How are we doing with explants, infections, things like that? Because this we know right away, even as we wait for the efficacy outcomes to accumulate over 7 or longer years. All right. And then resection. How do we judge seizure freedom? Well, all you have to, my suggestion is you just have to read the articles carefully. Is seizure freedom at last follow-up, which could be all over the map, 1 year, 2 years, completely seizure-free since surgery, and were there ASM changes? And typically there are. Just one example from the literature, and there's lots of literature on this, 160 adults with 15 years minimum post-op follow-up, so very long-term. Great news that almost 60% were seizure-free at most recent follow-up. That's not long-term follow-up necessarily, but at most recent follow-up, which may include, probably does include medication changes, and 31% were actually seizure-free for the entire duration. So when we talk about 50% or 70% seizure-free, if patients ask, well, what do you mean by that? You should have that data handy, that long-term, surgery, never another seizure, maybe more 30%, although this is certainly very encouraging and probably better than medication therapy alone. So all of these little details come into play when we're evaluating resection data. And to the fellows and early stage attendings on the call, you learn this stuff over time, but if you early in your practice, if you have a certain skepticism about some of the things, I think it's helpful. I think it can help you get a leg up as you're talking to your patients about the long-term outcomes of these things. So callosotomy, another interesting story. So there is a great expansion of corpus callosotomy, certainly in pediatrics. It is being used for almost any kind of presumed generalized seizure, including epileptic spasms. So as far as side effects, because I made a point to talk about safety and side effects, because you kind of know that right away. Good news. disconnection syndrome is absent or mild before age 10 years of age. That has been written multiple times, certainly been my experience. So kids do well after calisotomy from a cognitive behavioral point of view. It really, they might take a few weeks, maybe 3 months to get back to baseline, but then you really can't tell any difference. So that's the good news. So how well does it work? Well, using the 50% seizure reduction metric for the classic seizure type for a calisotomy, atonic seizures or drop attacks. So let's say 80% had a 50% seizure reduction, which beat VNS in this trial. But for epileptic spasms, which is the new favored seizure type for calisotomy, 77% had at least a 50% seizure reduction. And parents love to hear this, but use this with a grain of salt. Calisotomy for spasms, some are seizure-free. That sounds great. Well, you know what? I believe that to be true because 1954, Dr. Gibbs, writing about infantile spasms and hips arrhythmia. This is 1954. We have no treatments. About half the patients who are over 3 years of age had not had a seizure of any type for more than a year. The hips arrhythmic pattern also tends to disappear with increasing age. So you know what? I believe that we could do anything to some of our epileptic spasms patients, and some would be seizure-free because spasms go away sometimes completely. Seizures go away sometimes completely after spasms. So be skeptical about the intervention led to seizure freedom when data going way back when there were no treatments told us that some of these kids stopped having seizures. So far, we've just been talking about seizure frequency, which is obviously a very important outcome, but there's so much more. So we need to measure non-seizure outcomes in trials and in practice. Mood, behavior, sleep, attention, on it. There are validated scales for all of these. This is very doable. It's very hard in a trial to do this for many, many reasons. It's not for lack of trying that non-seizure outcomes are not trial measures. It's just really hard to do it. So a couple of shortcuts have been proposed recently. This is a very shortcut using the stoplight method, or called SIRIUS, or the Seizure Related Impact Assessment Scale. It was validated in adults with focal seizures, but it's being used in some pediatric DEE type trials. So what you do in adults, it was originally done monthly, but you could do it daily if you wanted to. So use this traffic light system to indicate how impacted you are. How many, let's say in a month or since the last visit, how many red light days did you have? You were unable to function because of seizures or side effects. Yellow light days, you could function partly, or green light days, you were fine. So this is on a monthly scale. So add up your red light, yellow light, and green light days, and you get something beyond just how many seizures did you have. A longer version, published also last year, was called the MORE scale, multi-dimensional outcome reporting in epilepsy. So this was a proposal, sort of an encouragement for us to try or think about trying something like this with our patients or in a trial. So there were different domains here. So the seizure control domain, so seizure frequency, seizure duration, and rescue therapy use. Depending on how well the patient's doing, they give the patients, this is self-reported, the patients give themselves points. More points are better. And then there's the quality of life domain. Did they feel cognitively better, emotionally better or not? Daily functioning, medication burden. The patient self-scores. Then there's the patient-reported experience domain, looking at these factors, patient self-scores, And the scale is if your total points are 90 to 100, then by this scale, you're doing well. And these categories are not necessarily important here, but imagine tracking this over time in our patients. Have them, in the waiting room or build it into Epic. I know that's a big lift. Build this in, and then we could track their overall multi-dimensional outcome reports in our charting. It's not been done, but it's an interesting, I think it's a thoughtful approach that we'll see where it goes. Okay, the last section. Precision therapy, because this has kind of been an old school talk up to this point, but so many exciting things are happening. So precision therapy, yeah, it's pretty straightforward. Well, not really, because each of these steps is many careers for many people. So how do we do precision therapy in epilepsy? Well, we unravel the biological mechanisms responsible for the clinical manifestations. Then we develop treatments that correct the molecular defect or its consequences. RNA modification, DNA modification, gene replacement, gene editing, small molecules, intracranial injections. And then we assess safety and efficacy in preclinical models. And then we do well-designed trials. And the trials look at impact beyond seizures on behavior, cognition, and development. So that's a big task, but it's actually happening in kids and adults. So any precision therapy trial has lots of questions to answer, like how young do you have to do it? How do you figure out what superiority is? How long can you have a blind? So let's look at some examples. Rett syndrome, Angelman syndrome, and SCN1A positive Dravet syndrome, not necessarily all Dravet syndrome. So you can cure anything in a mouse. I thought that was just something that some of my cynical colleagues said, but it turns out that's like it's a well-known scientific phrase that you can cure anything in a mouse, but translating to humans can be tricky. Well, all of these trials have very elegant animal models supporting their use. Rett syndrome. So gene therapy for Rett syndrome. Study is underway. And I bring this up mainly to show the outcome measure. And then we're going to look at the different outcome measures for different programs. So this is a single intrathecal injection of an AAV9 gene therapy. So it is a intrathecal times 1 dose. Patients get oral prednisone for 10 weeks. This is not pivotal yet. This is safety, tolerability, so there is no blinding or concurrent control group. It's possible in Rett syndrome they may never need it because their historical control database is so good, but we'll see. So the outcome measure is primary efficacy. Percentage of participants who are responders in the social/communication, fine motor, or gross motor milestones. So this study is taking a broad look. They'll take an improvement in any of these areas. Not every trial is doing that. I think part of the reason why a Rett syndrome study has the flexibility to look at such broad-based developmental outcomes is they have data going back decades on the natural history of Rett syndrome. So they know that social communication, fine motor, gross motor do not improve and actually regress slowly over time. So they'll take any improvement in any domain. They can probably do it without ever needing a control group. But that jury's still out on that completely. Angelman syndrome, all kinds of different precision therapy underway, many different sponsors, different phases. And this is one example. This is an ASO trial. So the investigational product is an ASO delivered intrathecally every 12 weeks. And there is a concurrent control group that gets placebo, artificial CSF, which actually makes the study much easier to do than trying to have a sham control group. So intrathecal every 12 weeks, and the outcome measure is expressive communication subdomain. You're going to see that again in another trial. Across multiple developmental epilepsies, parents, caregivers, there's a lot that there's many areas that could be improved. But if you ask many parents or caregivers, what if you could have one thing improved, what would it be? They will say expressive communication. So hence they are hanging their hat on improvement in expressive communication in the kids with Angelman syndrome. Now, you'll notice seizures are not, I haven't even mentioned seizures because they are not required for inclusion in the Rett and Angelman's arms. They'll be looked at from a safety point of view to make sure they don't worsen. But seizures are so hard to quantify and classify in Rett syndrome and Angelman syndrome that they're by design not looking at seizures as a primary outcome. And now Dravet. So various SCN 1A, 2A, 8A. We're going to focus on the SCN 1A protein for these trials. So there are gain of function mutations and there are loss of function mutations. There is some phenotypic difference, but there is also overlap. But Dravet, as I think most of you know, is viewed as a loss of function mutation due to SCN1A haploinsufficiency. And the two precision trials we're going to talk about are designed to correct haploinsufficiency. So what is haploinsufficiency? Well, you have two SCN1A alleles, and the mutant allele does nothing. There is no functional protein. The reason these trials work is that the wild type allele, by natural design, destroys 50% of the potential protein. So it's this natural destruction called nonsense-mediated decay of roughly half of the wild type product that is the basis for these programs, because the mutant allele remains mutant. So there's an ASO. Let's say there's an ASO that acts on the pre-mRNA. What does it do to the mutant? Nothing. Nothing. The mutant remains mutant, but it doubles the protein outcome of the wild type pre-mRNA. So that's why this works in animal models. So this is Stoke Therapeutics. And we know that the development of Dravet is heterogeneous, but we know that there are behavior and social emotional problems that emerge before 3 years and worsen. So there's enough natural history data to know that there are cognitive, developmental, behavioral challenges. So the ASO Dravet trial, which is in progress right now, phase 3 trial worldwide, intrathecal injections of the ASO every 14 weeks. There is a blinded concurrent control group. In the US, you get an LP with no artificial CSF. In the European Union, you get a needle prick. You don't get a full LP, a whole nother story behind that. But this requires a blinded and an unblinded study team, which is very complicated. Anyway, the point for this talk is the primary outcome is seizure reduction. The secondary outcome in the ASO trial is a change in baseline in Vineland outcomes in communication, daily living skills, socialization, or motor skills. So this sponsor is taking the broad approach as a secondary outcome. We're going to look broadly at development and see if this helps. There is another sponsor that is using not ASO trial, but what they call DNA regulation therapy, or for parlance, gene therapy, SCN1A gene therapy, that would basically have the same effect of doubling the output of the wild type SCN1A protein. So just like that cartoon I showed you before. Their study is different. It is a single intracerebral dose of this engineered transcription factor, DNA modification therapy, which they round up to call it gene therapy. Patients are immunosuppressed. There is a concurrent control group. The control group gets a sham burr hole in the operating room. but they don't get the injection. So there's a blinded and unblinded study team. And this trial, seizures are the primary outcome, and their secondary outcome is improvement change in expressive communication. So that's the second of these trials that have focused on expressive communication as their outcome. So you see a mix here. Some are doing broad-based developmental outcomes. Two of them in different diseases, Angelman and Dravet, are focusing on expressive communication at a year. So that was a lot, I know. Thanks for your attention. So we've covered where we are right now in 2026 with our natural history data, a little bit on devices. You heard my thoughts on treatment resistance, and we'll see what happens with precision therapy. And so think about expressive communication as being extra important to at least parents in kids with epilepsy. And I am happy to take questions if there are any.
Excellent talk, and we have multiple people giving you a round of applause. I'll open it to the floor.
Dennis, if I may, I have to particularly applaud you for talking about, as you put it, the normalization of deviance for neuromodulation. Is that the phrase correct?
Yes.
Am I using it correctly? This is something that has deeply concerned me for a bunch of years now, and I want to amplify your point. The thing that you didn't even get to, that it's gotten so bad now that there's a whole bunch of people in this field who are putting electrodes into the thalamus to try to optimize a therapy that is only maybe marginally effective itself. And I sometimes get the feeling that a whole bunch of people in our field have just completely lost the plot. I went to a symposium on this at the last AES to see if they could convince me that what they were doing was reasonable. I was pretty appalled by what I saw. So I'm thank you for, I didn't know anybody had published about this. So thank you for bringing that paper to our attention.
Dennis, I have a question about kind of technology and clinical trials, and with all respect to, I think we need to listen to parents, patients, and caregivers about outcomes, which I agree wholeheartedly. I think there's a lot of technology that has a lot of promise for helping us measure some outcomes, and the most of which is potentially going to be wearables, as well as also other technology to detect seizures. What is your opinion and some thoughts of how to weave in the utility in considering particularly the limitations of some of those devices now? Yeah, I think it's a little soon, but I think there's some significant promise.
Yeah, that is a great point, and I didn't address it at all in the talk. So from a trial point of view, many trials now use a wearable as some kind of exploratory outcome because they're just trying to figure out, like, can it work? And the FDA, there's a whole long discussion about what it would take for a wearable output to be actually a primary trial outcome. For years, there's been this research roundtable for epilepsy, and they had to spin off a separate device and wearable group just to talk about all of the issues with wearables in trials. In clinical practice, I'm really interested to hear the adult perspective. I think a pediatric colleague of mine in California routinely says, yeah, 2 to 4 weeks at most, and then the families throw them away. Like they're not sustainable for kids because of false positives and other things. Yeah, I think in practice it's feasibility. Like, how do we get patients to use them? We're going to switch to adult mode here. How do we get patients to use them? And then practically for us, like, what does the output data mean for me? Like when we'll hear from sponsors about, oh, we're developing this online technology to track this, that, or the other thing, and you can review, you meaning me, meaning us, you can review it in real time. I'm like, I don't want to do that. I don't want to see it in real time. Or, oh no, they'll bring it to the visit it? And then, well, wait, how am I going to do that? So there's, from the patient point of view, how do we make it usable for the patients? And then how are we either not overwhelmed by it or compensated for looking at it? So that's a long answer to your question, but yeah, that's an important topic.
No, it's okay. And actually, full disclosure, I guess I should have said before, I'm actually on that on that work group through the Epilepsy Foundation, and we're hoping to get some input from patients and caregivers in our April meeting, which is some of you may be familiar, there's a research ambassador program for patients and caregivers to learn a little bit about epilepsy and then give some feedback. So we're actually hoping to ask that question to them next month.
Great. Chris.
Dennis, thank you for an excellent talk as always. I think you kind of alluded to this, but I would like to get your thoughts. I think one of the challenges when we look at outcomes, and especially if we're looking at seizure outcomes, is we really don't take into effect the baseline seizure frequency, right? We all know in clinical practice, paradoxically, when somebody's having very frequent seizures and you start them on a drug, it's much easier to see if there's a response as opposed to that person that might have a seizure once every 6 months, once every 9 months. And I think that's one of the challenges in our field is when we talk about outcomes, we really don't think about how frequent some of these baseline seizures are. Do you agree that that's a...
I do agree. It's a great point, because if you have a seizure every 6 to 9 months, realistically, you're not going to be in a trial. So that's a clinical practice challenge. I think it leads me back to that Moore scale, the multidimensional outcome, because seizure frequency is only one of a whole range of options there. So yeah, absolutely. Pete.
Yeah, Dennis, thanks for a great talk as usual. Hey, I'm just wondering, when we talk about precision therapies, is it old-fashioned to still say that pyridoxine-dependent epilepsy and branched-chain amino acid dietary modification for MSUD still constitutes precision therapy, because they basically do target the molecular mechanism? So I just remember, because that was sort of like the paradigm that we always used. And I certainly, in the mTOR world, we thought that mTOR inhibitors should be precision therapy, but they are proving in clinical practice to be less, perhaps, precision than they ought to be, for sure. So in the field now, thinking about precision therapy, is the move basically largely just to correct the molecular ablation, or are there other strategies in the field for what constitutes precision therapy?
Well, so first of all, yeah, I agree. Those oral treatments of those epilepsies you mentioned, that is precision therapy. I mean, the cynical view of precision therapy is it won't work for almost anybody. It won't work for anybody else except you. So like that's like the reality, you know. So the definition in that paper I quoted about precision therapy was correct the molecular defect or its consequences. So it's basically any, and to be honest, I'm not a basic scientist, so I need a really slick cartoon to understand the mechanisms of these trials. And then, but yeah, they're trying all kinds of things.
I remain deeply skeptical of gene therapy. And again, the problem with it has got nothing to do with the molecular mechanism. We've fixed that easily. It's the delivery. It's getting it in. And an AAV9 is going to be fraught with, all different kinds of problems with it. On the other hand, if you look at ASOs such as SMA1, it was smashingly successful. So I guess we just keep pushing on.
Yeah. And we used to have this, in fact, we're at time, like the first time anyone mentions SMA in an epilepsy meeting, we're done. It's over, because it's a different disease. Totally. We are so unlikely to duplicate that success.
100%.
Yeah.
Thanks.
We are over time. Thank you for such a great and stimulating talk. That was the first time I think I've seen a round of applause from people in my time here. So thank you.
All right. Well, thank you very much. It was great to see you.
Thank you.
All right.
Take care.
Bye.